Welcome to Roadmap to Residency, Evidence-Based Applications. And in this video, we will be talking about T-cell development, the humoral and cell-mediated immunity. So when a cell is sensitized to the antigen on MHC1 or MHC2 comes into contact with a CD8 positive T cell or CD8 positive T cell respectively, the primary signal is merely the binding of MHC to both the T cell receptor and the CD molecule. So in other words, MHC1 binds to the T cell receptor plus CD8 and MHC2 binds to the T cell receptor plus CD4. A secondary signal that ensues, that is when the B7-1, that is CD80, or B7-2, that is CD86, on the nucleated cell or the antigen-presenting cell binds to the CD28 on the naive T cell, the newest T cell. The T cell is now activated. And in other words, the second step is the B7-1-2 binds to the CD28. And this second step is the same for both CD4 positive and CD8 positive T cells. So here is the drawing that I have made for you. So the antigen presenting cells, when the MHC gets bound to the antigen presenting cell, then there is the primary response where the MHC gets bound to the T cell receptor of the naive T cell. Okay, and again the CD28 of the naive T cell binds to the B7-1-2 of the antigen presenting cell, which provides the secondary signal. The activation of the T cell results in the CD8 positive cytotoxic cells directly killing the nucleated cells that have antigen loaded on MHC1. The CD4 positive T cells are able to exert a variety of effects depending upon their differentiation. The activation of the CD4 positive T cell usually occurs after its differentiation. So the summary of T cell activation is that the primary signal is when the MHC binds to the T cell receptor, and secondary signal is when the P7 protein binds to the CD28. So the T cell differentiation, so the TS0, TS naught cell, is an upstream type of CD4 positive T cell that has not yet differentiated. CD8 positive T cells are more simplistic and are merely activated to kill target cells, but CD4 positive T cells undergo various routes of differentiation in addition to their activation. Now, these all things we have discussed earlier in our previous videos. This is just a recap. Now the TH0 can become either TH1, TH2, and TH17 and T regulatory cells. TH1 cells stimulate the macrophages which kill the phagocytosed pathogens and organized into granulomas. The TH2 cells stimulate the B cells to become plasma cells, which make antibodies that kill the extracellular pathogens. The TH17 cells stimulate the neutrophilic production and recruitment, and the T regulatory cells prevent autoimmunity. That is, are a suppressive type of T cell. TS0 and TS1 cells occur via the stimulation. TS02, the differentiation of TS1 occurs via the stimulation with interleukin 12, interferon. So interferon gamma is secreted by TH1 cells. So once the TS0 cells becomes a TH1 cell, it activates macrophages via interferon gamma. Now the macrophages then secrete even more interleukin 12, which continues to stimulate more TS0 and TH1 cell. Now, these stimulated macrophages may or may not go to form granulomas, which are a dense cluster of organized macrophages that appear if inflammation is chronic. Now, the activated macrophages composing a granuloma are called histiocytes or epithelioid macrophages. Now, in short, we can say that interleukin 12 or interferon gamma will activate the TH1 lineage, which go on to form macrophage activation. So, the macrophages that organize into granulomas are called histiocytes or epithelial macrophages. Now, this is all we have discussed. The thing to remember is that TH0 to TH1 occurs via the stimulation of interleukin 12, and whenever there is a formation of TH1, then it activates the interferon gamma. Now, these activated macrophages will again secrete more and more interleukin 12, which will again form more and more TH1 from the TH0. So talking about humoral immunity. So humoral immunity means antibodies are involved in the killing of the organism. If antibodies are not involved, then the process does not relate to humoral immunity. Plasma B cells produce antibody and hence are the main cell of humoral immunity. The B cells are one type of APC that present antigen to TS2 CD4 positive T cells by MHC2. So the TH2 cells activate the B cells to become plasma cells, plus they induce the B cell isotype class. One thing you need to know is that the X-linked A gamma globulinemia and IgA deficiency are deficiencies of humoral immunity, very frequently tested in the examination. Talking about cell-mediated immunity, which means T-cell-mediated immunity that does not involve antibodies in order to kill the organism. Cytotoxic CD8 positive T cells kill the nucleated cells that present intracellular antigen and MHC1 molecules, where the CD8 positive T cell releases the granzymes and granulysins and perforins, inducing apoptosis of the target cell. TS1 or CD4 positive cells activate macrophages via the interferon gamma and interleukin 12 loop to kill the phagocytous organism. So the dendritic cells are another type of antigen-presenting cells that are part of both humoral and cell-mediated immunity, depending on if the MHC2 TCR interaction results in a TS1 or TS2 response. So TS1 is a cell-mediated response and TS2 is a humoral response. So talking about the cell-mediated immunity steps, so first the MHC1 binds to the T cell receptor on CD8 positive T cell. And then the B7-1 or 7-2 on nucleated cell binds to the CD28 on CD8 positive T cell. Then the activated CD8 positive T cell induces apoptosis in infected cells by releasing granzymes and granulysins. So most of the viruses and bacteria like Listeria uses this cell-mediated immunity pathway, also the Mycobacterium species. Now, this is the simple flow chart of the humoral immunity that I have found. So, antigens are produced from pathogens or the patient and circulate in the plasma, lymph, or other tissue. Then, either it may be a T cell dependent antigen or it may be a T cell independent antigen. So, antigen presenting cells present the antigen to CD4 positive helper T cells and activate them. Whereas in case of T cell independent antigen, the antigens such as peptides, carbohydrates, or lipids may be directly recognized by the B cell. Then they go on to act on the naive B cells to form activated B cells. Now these activated B cells undergo clonal expansion to either increase the WBC or may go to act as an autoimmune disease. So if the B cell recognizes the antigen, the self antigen as a foreign antigen, then the clonal expansion will lead to the differentiation of the B cells into either memory B cells, plasma cells, which can produce antibodies and all. Thank you for watching. Keep studying.